Hello, we're glad you've joined us for this live webinar, Isolation and Expansion of Mesenchymal Stromal or Stem Cells for Translational and Clinical Research. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Miltenyi Biotec. Miltenyi Biotec is a global provider of products and services that advance biomedical research and cellular therapy. The company's innovative tools support research at every level, from basic research to translational research to clinical application. This integrated portfolio enables scientists and clinicians to obtain, analyze, and utilize the cell. The company also offers comprehensive scientific support, consultation, and expert training. To learn more about Miltenyi Biotec, visit www.miltenyibiotec.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Kathrin Godthardt, a team coordinator and project manager of the R&D Stem Cell and Regenerative Medicine Group at Miltenyi Biotec. Kathrin joined Miltenyi Biotec's R&D department in 2001. She received a bachelor's degree from the University of Mainz in Germany and finished her thesis on characterization of endothelia progenitor cells at Miltenyi Biotec. Kathrin has long-term experience on separation technologies, development of cell culture media, and adaptation of separation and cultivation processes into clinical-scale closed systems for mesenchymal stem cells. Kathrin Godthardt will now begin her presentation.
Hello, everybody. Thank you, Judy, for this nice introduction to this Miltenyi Biotec webinar, which is on the isolation and expansion of mesenchymal stem cells for translational and clinical research. So this is here now the presentation of our disclaimer. We are providing several research reagents, and some of them are FDA approved. So for more information, you can here find a lot of interesting information. So the agenda for today is depicted here. So first, I would like to give you some interesting facts about Miltenyi Biotec. Furthermore, then we come to our topic, so clinical scale expansion of MSCs. I would like to introduce our serum and xeno-free cultivation solution and also our new Clinimax Prodigy adherent cell culture system. I will end my presentation with a brief summary. So first here, I will come now to some interesting facts. So Miltenyi Biotec was founded in 1989, and now we have a number of employees, so up to 2,500 people are working for Miltenyi Biotec worldwide. We are from more than 44 nations. I'm part of the R&D department, so this group is quite big, so 20% of all the employees are working in our R&D department, and we have worked on about 20,000 scientific publications up to now, describing a lot of interesting work in all our different fields we are working. So the product portfolio is also quite bright, so more than 15,000 products are available, and a lot of our products are in the cell therapy field, so clinical field, more than 55,000 cell therapy or cell therapies were produced with Miltenyi Biotec products. About 2,500 patients are treated up to now per year with the use of our technology. Our products are available in 28 countries all over the world, and we have 73 countries which are distributed by several distributors. So our aim is to make it easy to have some research on the one hand and go into the clinic later on. So from bench to bedside, this is what we would like to do. And so in this case, we are providing sample preparation reagents, reagents for cell isolation, activation, and expansion, of course. And after expansion, cells can be analyzed using our analysis products and also the flow cytometer. If the work from research goes more and more into preclinic or into the clinic, we make it quite easy with our bench-to-bedside aim, and therefore here we have several products which are very similar in research or in clinic. So for example, the cell culture media are very similar, so it's very easy to cultivate cells afterwards for clinical applications, cell analysis, and also cell processing with the prodigy. So now I'd like to point out challenges in clinical scale expansion of MSCs. So here briefly, there's the MSC workflow, which we are following, or a number of people in the MSC field are following. So first, we start with the starting material. Different kinds of tissues are used: bone marrow, adipose tissue, umbilical cord. MSCs are then isolated by plastic adherents and expanded depending on the application you are following. So on the one hand, of course, small-scale research applications are used, and also a lot of clinical applications, small-scale, when you think about autologous settings, or large-scale expansions for any kind of allogeneic clinical settings. After expansion, in most cases, cells are cryopreserved and quality controlled. So this workflow, of course, bears a lot of challenges, and there is a nice and informative survey from RecMedNet on current trends and future developments in MSc research. This survey was available to all members of the RecMedNet community and was completed by scientists, students, technicians, group leaders from both academia and from biotechnology and medical companies. So here it comes out that the biggest challenges were identified in transferring processes to GMP and also to maintain the phenotype and the function of MSCs. So what is needed to overcome these challenges when working with MSCs? And here also the survey gave some nice feedback. So most important for people in the field are improved cell culture products, better methods of expansion. People need help in complying with GMP regulations, and also it is quite important to have integrated closed systems. So now I'd like to show you how Miltenyi Biotec, with our product portfolio in the MSC field, tries to meet these challenges and tries to improve the workflow for our customers here in this field. So coming now first to the cell culture media, so the xeno and serum-free cell culture media. For a long time, we are providing FCS-containing cell culture media now. But after the field more and more goes into clinic, xeno-free media are more and more important and coming into the focus. So therefore, we are providing xeno and serum-free media now. So there is the StemMax MSC Expansion Media Kit XF, which is a research-grade media. It's provided in 500 milliliter bottles with a corresponding supplement. Of course, it's xeno and serum-free, the whole formulation. No substrate for pre-coating of cell culture vessels is required, so this is quite a unique point here. Because in a lot of other cases from commercial available media, a substrate is necessary. So in this case, it's not. Also, when you start with primary tissue. And also, there is no additional supplement, growth factors, or any other kind of serum necessary to start your MSC cultivation process. And now, if you would like to go more into the clinic, focus more on clinical, preclinical work, we are providing the MSC-Brew GMP medium, which really shows the same formulation, the same benefits like the StemMax MSC Expansion Media XF, only the reagents or the raw material are regulated to a higher grade to meet the GMP standards. So this media, the MSC-Brew media, comes in bags, so in 500 milliliter or 2 liter bags. It's phenol red-free. We are also providing corresponding supplements which are aseptically filled. And the whole formulation, as I said, is in compliance with relevant GMP guidelines. During the development, we followed the recommendations of USP 1043 on ancillary materials. So this medium is quite a nice solution to start GMP-compliant work and to go further into the clinic. So now I'd like to show you some important facts regarding our medium with a perspective of performance. So the StemMax MSC XF and MSC-Brew GMP media support colony formation starting with primary tissue. Here on this slide you can see the number of colonies per well in bars, and the dots are depicting the cell number per well. So we have compared here our MSC XF medium and the MSC-Brew medium with a standard used homebrew alpha mem or DMEM with platelet lysate and FCS. And furthermore, we have compared to competitors where, of course, here in this case, you really have to add a B serum for this first isolation of cells from the starting material. So it's not possible to use the competitor media here without a B serum. There are no colonies at all at the end in the cell culture well. So better MSC adhesion results in a higher number of CFUF colonies using the StemMax XF and the MSC-Brew GMP media. But of course, we were also interested in how efficient the clinical scale isolation of MSC is using our cell culture media. Again, here we compared the XF and the MSC-Brew medium with the DMEM and alpha-mem homebrew media. We used 3 different donors, 10 milliliter bone marrow aspirate each. We cultivated the cells until we get 200 million MSCs, which is round about the number which is used for autologous clinical applications. And we could show you quite nicely that just after 15 days, we are harvesting this 200 million of cells using our media, the StemMax and the MSC-Brew medium. So when we use the DMEM or alpha-mem containing medium, this took much longer, so more than 30 days. Several passages were necessary, and even for the SCS-containing medium, for 2 donors at all, we could not reach the number of cells after 30 days. So here we have some pictures showing that we are quite fast with our medium. So just in 14 days, we are reaching the 200 million MSCs. And of course, when you are faster, you are not using so much medium. So the medium consumption is also reduced from 50 to 70%, which of course reduces some costs here. But not only the cell number is important, it's also important how the cells are behaving after long-term cultivation. So in this case here, we show some data expanding MSCs from bone marrow for about 6 passages in the 4 different conditions. And as you can see, there is a stable growth rate for the StemMax XF and the Mse-Brew GMP medium. So after expansion, the MSCs were analyzed for their phenotype and their function. So in this case here, we were using the MSC phenotyping kit, which is a flow-based assay to analyze surface expression markers. The MSC phenotyping kit consists 3 different positive markers, CD105, CD73, and CD90, and also some negative markers, CD14, CD20, CD45, and CD34. These are all markers which were also mentioned from the ICT consortium to be used for MSC identification. So this assay is quite reproducible and rapid, and it's a cocktail, so it's quite easy to use. Furthermore, it's important that MSCs show their multi-lineage differentiation potential, and we have analyzed this by using the Miltenyi differentiation media for adipocytes, osteoblasts, and chondrocytes. The pictures are here shown in the middle, and as you can see, the differentiation potential is observed after the 6 passages for when you use the Mse-Brew GMP medium or StemMax medium. And also, we looked for a specific function of MSCs, which is related to their immune modulatory capacity. So here we use the T-cell suppression inspector, which is an assay to analyze the T-cell, the suppression of proliferating T-cells. So to explain this in more detail, T-cells were labeled with a membrane dye, and then within a flow-based assay, after stimulation, you can detect the proliferation rate of T-cells by the dilution rate of the membrane dye. And when you then spike MSCs in different concentration and co-culture them with T-cells, you can really nice detect this suppression of T-cell proliferation. And this works quite nice in all cases, whether you use the Mse-Brew GMP medium or the homebrew alpha-MEM and DMEM cell culture media. So coming now to this new Clinimax Prodigy adherent cell culture system. So here you can see again the quite complex way of producing cells in a GMP-like manner. So there are different steps. There are a number of different devices you have to use. So incubator, centrifuge, you have to use microscope, and a lot of hands-on time, of course. Everything is done in clean room. It's really not easy to go in and out without spending a lot of money, a lot of time. So it's quite a time-consuming process, and our aim here is to bring everything together in one solution, so the Clinimax Prodigy as an all-in-one solution for the complex cell product manufacturing. So now, if we would like to have a closer look, the system looks like that. So here we have the hardware of the Clinimax Prodigy platform. We have an integrated and closed system, so where you can really do a lot of automated cell processing steps. For example, the sample preparation, density gradient centrifugation is done within the centrifugal unit, which you can see here on the right side. Cell washing can be done, media change, magnetic cell separation, cell stimulation, genetic modification, and of course, at the end, the final product formulation. So the whole system is closed from sample to product, and this is done by single-use disposable tubing sets, the sterile filters and input ports, and there is the possibility to do sterile welding in a lot of cases, for example, to connect our cell culture media bag sterile to the tubing set, to connect cell cultivation vessels, so everything can be connected in a sterile way. There are integrated QC sample pouches where you easily can take samples during the cell cultivation process to analyze the cells between different passages. So I mentioned the centrifugal unit, so the CCU, and also external culture vessels. Now I'd like to introduce this in more detail. So the centrifugal unit, the CCU, has got a surface of 100 square centimeter. This can be used for the cultivation of adherent and non-adherent cells. Of course, for MSCs, this is not really a large area, so therefore later on I will come to the external culture vessels. But of course, when you think about starting with umbilical cord or small tissue pieces which are enzymatically digested, then it could be an option to use this 100 square centimeters. So to really start with a small scale and then upscale this. So this is possible here in this CCU. So it's also possible then to have the automated feeding and harvesting procedure with the centrifugal unit. It's temperature controlled from 4 to 38 degrees. Also CO2, air, and N2 controlled to get a sufficient environment for cell growing. We have got a microscope integrated here in the Prodigy for visual cell examination. And of course, multiple input ports for different media or cytokines. But of course, if you like to cultivate larger surfaces, which are really required for the Mse workflow, there is this really nice possibility to connect in a sterile well external culture vessels. And this gives you the opportunity for a really scalable cell manufacturing process. So coming now to the tubing set, the closed tubing set, which is used for this cultivation process. So it's a GMP-compliant tubing set, where you have sterile welding possibilities, as I already said. So you can connect the cell culture media, of course, everything else which is also necessary. So the enzymatic digestion solution, the enzymatic stopping solution, the integrated bags and sample pouches, as you can see on the in the middle of the picture, where you can the QC samples during the different passages, and you have the easy connection of the external culture vessel here on the lower level of the tubing set at valve 19. For cell culture vessels, we are using here in this picture, as you can see, cell stacks which are available in different sizes from 1 layer, 2 layer, or 5 layer. And at the end, you can connect 3 different stacks in parallel and can process them subsequently with a prodigy. Which is also possible is to connect the Clinimax electroporator to this system, which then gives you the possibility for genetic modification of cells. So here you can see the GMP-compliant MSC expansion process, how we have developed them, this process for the Clinimax prodigy adherent cell culture system. So everything is done as you would also do it manually. So you start with bone marrow, you have the density gradient centrifugation, which is performed completely automated in the device. Afterwards, you can harvest the bone marrow mononuclear cells, which are washed in the cell cultivation media, so that afterwards you can directly start the cultivation in the external cell culture vessel. Cells are splitted in this closed system, again seeded in higher or larger surface area, cultivated again up to the number of passages which are necessary for your process. At the end, you can harvest the cells automatically, choose a sufficient harvest medium, which you might also use for direct application or freezing. And at the end, you have your final cell product. Of course, this process is also adaptable to different starting materials like umbilical cord and adipose tissue. So here there is a nice picture which shows the performance of our process. So we started with 30 milliliter of bone marrow. The whole process can be processed with 30 up to 100 milliliter of bone marrow, but in this case here we show the example of 30 milliliter. We started with the density gradient centrifugation to yield bone marrow mononuclear cells. We use the MSC-Brew GMP medium and compared then the expansion of cells with the manual process. So in both cases, after 10 days, cells were around about 70 to 80% confluent. We harvested cells in the Clinimax prodigy by starting the harvesting procedure, seeded the cells again, and ended up then after 14 days with 400 million MSCs, which is quite a high number. So at the end, we used 3 5-layer stacks in parallel, processed by the Clinimax prodigy. So of course, the characterization of MSC is also very important. So again, here we use the MSC phenotyping kit, including the analysis for CD73, CD105, and CD90, and all markers were highly expressed, as well as the MSC, the non-MSC markers, which were negative on these cells. So roundabout at the end, we also calculated the time we were using in both cases, so the manual process and the Clinimax process. And here the graphic on the right side shows quite nicely that we could reduce the hands-on time using the prodigy for about 70%. So this at the end is quite interesting. So because all the hands-on time which is done in the clean room can be really reduced to a minimum. So coming now to the summary, here we talk about the challenges which came up in the clinical field to process to transfer the process to GMP, for example, and which is or what is really important at the end for the people in the field is the improvement of cell culture products, better methods for expansion, helping in complying with GMP and having integrated closed system. And for all these things, we could show quite nice solutions by introducing our xeno and serum-free cell culture media, and also to provide the closed adherent cell culture process using the Clinimax Prodigy. So the improved serum xeno-free medium supports efficient clinical scale MSC expansion from different tissue sources. The new Clinimax Prodigy adherent cell culture system allows MSCs to isolate and expand in an integrated closed system. And we have complete workflow for standardized MSC characterization, isolation, expansion, phenotyping, and functional analysis. So thank you very much for your attention.
Thank you, Kathrin, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Kathrin Godthardt will answer as many questions as time permits. The first question is, how many MSC cells can you acquire from the Accc process Prodigy using 3 5-layer cell stacks? And is it possible to include more cell stacks to acquire a higher number of cells?
Okay, so with 3 5-layer stacks, we can harvest around about 400 million MSCs, which is quite a high number. And of course, it's also possible to upscale the process to 6 5-layer stacks. So that would be related to a surface of nearly 20,000 square centimeters, so which is about 2 square meters. And we are planning some experiments in the near future to test this for MSCs from bone marrow and some other tissues in the near future.
Thank you. Next question is, can you explain what are the differences between your GMP medium and research-grade XF medium?
So the formulation of the MSC expansion medium XF and MSC-Brew GMP medium is really the same. So however, these two products are produced in different manufacturing sites. So MSC expansion medium XF is produced in Bergisch Gladbach, and the MSC-Brew GMP medium is produced in our GMP facility in Teterow. Since the production processes of these two media slightly differ, finally products have derived different formats. So one supplement versus 2 supplements, for example. So 1 supplement in the XF medium and 2 supplements in the GMP medium. Also, the GMP medium is provided in very convenient bags, 500 milliliter and 2 liter, which makes it easier to stay in completely closed environment during the cell processing. So furthermore, our GMP media are manufactured and tested under quality systems certified to ISO 13485 and ISO 9001. So the design of the media follows the recommendations of USP 1043 and is in compliance with relevant GMP guidelines. All media are filled using an aseptically filling line. All media undergo a stringent quality testing, including functional quality controls, as well as, for example, sterility testing according to the standards of the European Pharmacopoeia. So the GMP media we are providing require documentation for regulatory review, of course, and we are including the certificate of analysis, certificate of origin, and the product information file.
Thank you. The next question is, can you comment on the differences between the Accc process in Prodigy and other bioreactors, for example, Quantum? And can you explain a bit more about the benefits of the Accc process in Prodigy compared to other bioreactors?
Yes, so with the Prodigy, we have a scalable system. So as I explained, there's a centrifugal unit with 100 square centimeter, and we can upscale the cultivation area to 9,000 square centimeter or even 20,000 square centimeter when we use 6 5-layer stacks. While in other bioreactors, they have fixed area, therefore they are not able to scale up the process. So, and that can be quite not so easy when you start with small starting volumes of material. So, for example, low number of bone marrow, low number of adipose tissue. So therefore, it's not so flexible, and in our system, everything is quite very flexible. So furthermore, we do not need to coat the cell stacks for the culture, while in most cases other bioreactors, so whether you use carriers for cultivation or hollow fiber modules, they need always to be coated before you start the cultivation, whatever medium you are using. So this is quite time consuming, and this is also quite expensive at the end. So in this case, there's quite some advantage in our system. So furthermore, the density gradient centrifugation process is included in our ACC process on the prodigy. So therefore, we have less contamination of other cells, and cell proliferation is more efficient. Of course, you can also do the density gradient centrifugation manually, but of course, for example, 100 milliliter of bone marrow, this really takes long. So a closed and automated system is quite has quite advantages here. So other bioreactors don't include the DGC process. Therefore, the starting culture cells contain many other cells, for example, erythrocytes, and of course, the proliferation is not very efficient in this way. So furthermore, the growth and the detachment can be observed partly under the microscope when we use the prodigy process. Therefore, we have better control of cell growth and harvest. So other bioreactors are not able to observe these under the microscope. For example, when you think about hollow fiber modules, or also when you use a carrier system, the harvesting step is also always a little bit tricky here.
Thank you. The next one is the ACC process also applied for umbilical cord or adipose tissue?
Yes, so it's possible to use the ACC process also with these starting material. Of course, so the enzymatic digestion of umbilical cord and also adipose tissue has to be done manually, so this cannot be done within the process. But after you have your single cells, after enzymatic digestion, you can enter the prodigy process and also start the whole cultivation process.
Thank you. To what extent can the ACC processes be automated?
So when the liquid, for example, the media or the buffer is pumped out or pumped into the stack, you just need to turn the stack so that the liquid can be pumped out or evenly distribute in the stack. This takes only a few seconds. Other than this, the whole process is done automatically. So you can save around about 70% of hands-on time compared to the manual system. And also the whole DGC process is completely done automated. So you can save a lot of time here.
Great. Has the prodigy system already been used in any clinical studies or cell therapy trials?
So the prodigy has been used for several clinical trials in the field for graft engineering or for TCR alpha beta systems. So a lot of things have been done here, and of course you can find a lot of more information on our website.
Thank you. Next one is, does your MSC expansion medium work well also with MSCs from umbilical cord or adipose tissue?
So the MSC-Brew GMP medium and also the StemMax XF medium can be used also with umbilical cord or adipose tissue or from starting material, and also just to expand the cells. And there's also no adaptation time necessary when you switch between different cell culture media.
Okay. Next one is, what markers do you include in your MSC phenotyping kit?
So the MSC phenotyping kit includes CD73, CD105, CD90. They are the positive marker, and it's including a set of negative marker, which is CD14, 20, 34, 45. The cocktail does contain PE, FITS, APC, and PerCP. And so it's possible to also combine some other markers with a cocktail, for example, HLA-DR. So all these markers in combination go in line with the criteria defined by the ICT consortium.
Okay, it looks like we have time for one more question. That will be, can you explain a bit more on how your MSC immunosuppression inspector works?
Yes, so the suppression inspector is a kit to analyze the immune modulatory capacity of MSCs. So the reagent consists of CD3, CD2, CD28 coated particles, which are used to stimulate the T cells. So the T cells are previously labeled with a membrane day, and that means then after stimulation, the proliferated T cells can be analyzed via flow cytometry, and here the dilution of the membrane day can be detected. So when co-culturing the T cells now with MSCs in different dilutions, you can observe a reduced T cell proliferation. And this is what you would like to see, because this is the effect of the immune modulatory capacity of MSCs.
I would like to once again thank Kathrin Godthardt for her presentation. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. The LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.